Hi, so my name's Amy Cooper and I'm a PhD student at the University of Aberdeen. And I'm about a year and a half into a 4-year PhD programme with East Bio, who are a funding body that fund doctoral training programmes around the east of Scotland. So predominantly Aberdeen, Dundee, St Andrews and Edinburgh. This is the first talk I have ever done of this nature, so I've definitely never delivered a talk from the floor of my spare bedroom in my little Aberdeenshire flat, and I've never delivered a talk to my wall before. But I'm looking forward to this new challenge for National Insect Week. So before I tell you about my PhD project and what I study, I'm going to tell you a little bit about how I got here, because it definitely wasn't the normal route into academia. So I grew up in Hereford in the West Midlands, and I left school at 16. So I got out of there as soon as I could. I hated school. Although I did enjoy learning, but I just didn't think I was clever enough to follow my friends to university. So I worked as a cleaner and a barmaid and a housekeeper between Hereford and North Yorkshire until I was about 23, I think. And throughout that time, I had lots of fun. I earned enough money to go on lots of adventures and do lots of travelling, which was great. But after all that, I kind of thought perhaps it was time to try and get a proper career. So I joined the Merchant Navy, which probably seems like a bit of a curveball. The Merchant Navy gave me my first taste of living in Scotland, which I loved. Unfortunately, what I did not love was the job. It was just not a good fit for me at all. But what was fortunate is that it gave me the skills to actually get a job with the RSPB. So I managed to get a job working in conservation on Ramsay Island off Pembrokeshire, and I loved that so much that I then found a position on the Isle of Islay over winter and then returned to Ramsay for the following summer. So throughout my conservation experience, I had the opportunity to work with lots of different scientists who were studying lots of different things. And I was just able to chat to them about their lives and help them collect their data. And I really found them inspiring and looked up to them. And as much as I loved conservation, I loved the practical work, I didn't necessarily aspire to the career progression that was available to me within conservation. So I thought, actually, perhaps I'd like to work more behind the scenes at gathering data and science that could contribute to helping wildlife and nature. So I thought I would see about getting a degree. So I was very lucky in that the Aberystwyth University in the west of Wales took me on regardless of the fact that I didn't have A-levels, and took me on the strength of my conservation experience. But I was definitely a high-risk student for them. So I loved it. I absolutely thrived. And it was a great 30th birthday present to graduate with my bachelor's degree. So after my undergrad, I decided that I wanted to carry on and do a master's. So I moved further north and across the border to Chester. And pretty much as soon as I started doing my master's, I started to scroll through findaphd.com to look for my perfect PhD, which I found up here in Aberdeen. So before I tell you what that perfect PhD is, I just want to say that doing a PhD is absolutely a team sport. There is no way I could do this on my own. So I have 3 supervisors. So I have Dr. Alan Bowman, who's based at the University of Aberdeen. I have Dr. Ewan Campbell, who's at the Centre for Genomics, which is actually in the Botanical Gardens, part of the university. And Professor Tom Freeman. So I think unfortunately my face covers his, but he's over in the Roslin Institute at the University of Edinburgh. So I'm also part of an ever-growing lab group. So our lab group predominantly studies honeybee health, but actually Antonio, who's new, he studies ticks and Lyme disease, and actually now he studies a little bit of COVID-19 as well. So because of this pandemic situation, lots of researchers are now turning their skills to help the efforts to get us all safe and back out of lockdown. So all of these guys are based in Old Aberdeen, where I spend most of my time. But on the left, we have the microscopy and histology department, who are based over in Forester Hill near the hospital. So I have the great opportunity to work alongside them and produce beautiful images, many of which are actually featured in this talk. So great, finally, what is my PhD? So the title is Elucidating the Honeybee Immune Response and Pathogen Transmission at the Varroa Mite Feeding Site. And essentially what that means is that I look at the interactions between 3 different species. The honeybee, which is featured on the top left, the Varroa mite, which is featured top right, and the deformed wing virus, the effects of which you can see in the bottom picture. And I'm just going to tell you a little bit about those organisms individually now. So I'm sure you're all aware that honeybees are incredibly important. In fact, they are our most economically important managed pollinator, and they pollinate many of our favourite foods, so things like avocado. And they also help wild pollinators maintain floral biodiversity. And I'm sure you're also probably aware, because luckily for honeybees, they have great media coverage. So you probably know that they're undergoing a major health crisis. So beekeepers often lose about 30% or 1 in 3 of their hives every winter. And that's because of lots of different reasons. So things like increases in intensive agriculture, so the rise in the monoculture, changes in beekeeping practice, such as the large-scale migratory beekeeping that you see over in America, where hives are driven from one end of the country to the other to pollinate different crops, so things like almonds and apples. But also problematic to honeybee health is the thing that I'm interested in, and that's pests and pathogens, so largely the Varroa mite and the deformed wing virus featured here. So what is the Varroa mite? I'm sure this is a little bit more unfamiliar to most of you. So the Varroa mite originated over in Asia and infested the Asian honeybee, but it moved west as part of the global honeybee trade. And it was so detrimental to European honeybees because they just weren't used to this threat. They didn't have well-established ways of defending themselves. And one of the major problems is that Varroa mites in Asia only affect the drone bee. So the drone is the male, whereas in Europe and, well, the rest of the world, in the European honeybee, the Varroa mite affects both the worker, the female, and the drone, the male, and they have very different roles. So the sole purpose of the drone is to mate with the queen and fertilize her eggs. And once that's done, you know, he's pretty much finished. But the worker bee, she does pretty much everything else around the hive. She cleans it, she raises the brood, and she goes out to forage for food. So as you can imagine, having both drone and worker affected in the European honeybee is a real problem. So how does the Varroa mite reproduce? So the queen bee comes along and she lays an egg, and that you can see in the drawing on the left. And that egg develops into a larva, which is depicted just next to the egg. And that larva is originally in an uncapped cell. So these are cells that are open to the air in the hive. So at some point, that larva releases pheromones that tell the worker bees to come and cap the cell. So you can see these nice waxy caps next to the open honeybee cells here. So when the larva is releasing those pheromones, that's also the time that the Varroa mite sneaks inside the cell and hides. And as soon as that cell is capped, she emerges from hiding and starts to raise her family. So what she'll do is she'll first lay an unfertilized egg, which becomes the male, and then subsequently she'll lay fertilized eggs, which become females. And then within the cell, that male will mate with its sisters, and then that whole dysfunctional family will feed on the developing honeybee. And they will feed throughout development from larva to pupa until the bee is ready to leave the cell. And what they'll do is they'll feed from a single feeding site, which is pictured here. And they'll continually return to that feeding site and keep it open and keep feeding it. So I think of it a little bit like a watering hole in a savanna that all the animals kind of come around and drink from. So as you can imagine, that's detrimental to the honeybee's health on its own, but it's also a great site of pathogen transmission. So the Varroa mite continues feeding until the honeybee is ready to leave the cell. And throughout that time, it's able to transmit deformed wing virus, as well as other pathogens. But we'll stick with deformed wing virus, seeing as that's what I work on. So the deformed wing virus does what you can imagine it does from the name. So it deforms the wings, as shown in this picture, and it also gives the bees this shortened, bloated abdomen. And aside from the physical effects, it also lowers the survival rate for honeybees. And as you can see here, like this honeybee is not going to be able to function properly in the hive. It can't fly. So what do I do to study these organisms and the interactions between them? So a large part of my PhD is actually genetics. So I look at gene expression differences, essentially in honeybees that are infested with Varroa mite compared to those that aren't. But I'm not going to talk about that today, because I think what's probably more interesting and definitely far prettier is the histology work that I do with the microscopy department over in Forester Hill. So the first thing that you always do when you start a new project is, of course, read, and I was no different. So I wanted to find out what images had already been created of the Varroa mite feeding site. And it turns out not many, which is surprising, seeing as the honeybee is so important. And the interactions between the honeybee, the Varroa mite, and deformed wing virus are so crucial. But I did find these two groups that have both pictured the feeding site. So on the left, we have Cambar and Engels, and their bottom two images are of the feeding site, and it's depicted in two very different ways. So on the bottom right, we have a very deep hole, and on the bottom left, we have a partially healed wound site with hemolymph spilling out of it. So hemolymph is the equivalent of bee blood. And then above those images, Cambar and Engels found two distinct different types of bacteria within the area of the feeding site. And one of those is another honeybee pathogen, which causes European foul brood. So another really terrible honeybee disease. And similarly, on the right, Ramsey et al. Found two different types of bacteria underneath the wound, but didn't specify what these were. And they also found partially digested fat body underneath the wound site. And this adds to a larger question about what the Varroa mite actually feeds on. Does it feed on hemolymph, bee blood, or does it feed on fat body, which is the bee equivalent of liver? Or does it feed on a little bit of both? So both of these groups worked on bees that were much older than mine. So I work on quite young honeybees developing pupa. So the first thing I needed to do was figure out what works on my samples, what kind of images can I produce, and what kind of questions can I ask. So this felt like the natural and simple place to start. So this is using a technique called bright field microscopy, and it's using a microscope that you're probably most familiar with if you've done any microscopy at all, and that's the kind of microscope that's pictured on the bottom right, although of course the ones at the university are far bigger and fancier and hooked up to computers than this one here. So I guess to begin with, I appreciate that I'm going through a lot of different microscopy techniques, but I don't think it's super important here for me to describe exactly what they are. I think that would take too much time, and to be honest, I'm still learning myself. And also I appreciate that some of these images are quite abstract. So I have included some little images like the one on the bottom left, which show where the feeding site is on the bee. So in this case, it's on the upper abdomen, and the feeding site is this blue fuzzy ring. And also in terms of what you're looking at in the images themselves, the oval shapes in the top right of the histology pictures, cross sections of the legs, and then everything on the left of the picture is abdomen. So this is what I think is the first section or cross section I have through the Varroa Mite Feeding Site, which was super exciting because these were really difficult images to get. They certainly weren't simple or easy. And quite frankly, that's just because the honeybee pupa is very squishy. So as I might have already mentioned, they're just sacs of hemolymph and fat body. So it took us quite a while to learn the appropriate methods to fix them or make them sturdy enough that we could section or slice through the bees. But I think all that hard work just made it more exciting when I thought, yes, this is the first picture I have of the Varroa Mite Feeding Site. But although I was excited, I still had a slight problem, because how did I know that this was definitely the feeding site and not some artifact of processing or some damage that I caused when I was removing the pupa from the cell? How did I know that I hadn't just picked the feature that just looked the best on my slide? And I think the answer to that comes from just a happy accident. So everything has a natural fluorescence. The bee here has a natural fluorescence of green, and luckily, so when I find the feeding site, what I do is I stain the bee with something called trypan blue, which stains the dead cells around the feeding site blue, funnily enough. And that stain fluoresces differently from the rest of the bee. So I have the feeding site within the red circle, and then the arrow points to this yellow section, which I hope is visible on your screens. And that yellow fluorescence is around the area that I think the feeding site is. So I can match these two images up, and I can actually superimpose them as well. So that's incredibly helpful. And I'm really looking forward to getting more sections like this when we're eventually allowed back to study in the labs. So this is just a zoomed in image of some of the pictures that I've already shown you, and just gives you more of an idea of detail. So on the left, we have the cuticle, so sort of the skin of the bee. The dotty cells are all lipid droplets. So like I've already mentioned, the pupa is just a little bag of hemolymph, bee blood, and lipid or fat body. And then these cells are oonocytes, these really dark cells. So actually, we don't fully understand exactly which cell types are within the honeybee, and the function of the cells that we do know exist hasn't been fully explained. But what we do know is that the oonocytes have a role in the immune system, and that they are part of a wound healing response that's caused by melanization. So with this information, I can start to ask questions like, are there more oonocytes in bees that are infested with Varroa mite than those that are not infested? Are there different immune cells that I can see in bees that are infested with Varroa mite compared to those that aren't? And are these immune cells attracted to the wound site itself, or do they just like float freely around the body? So as I mentioned, it was really difficult to get those sections, and what we thought might be easier was to digitally section the bee instead of physically section it. So we looked at using micro CT. So if you've ever been to hospital and had a CT scan, this is essentially the same, although an awful lot smaller. And we even use some of the same contrast stains that are used in humans, and this bee is stained with iodine. So this gives us the ability to take lots of different sections from lots of different angles, some of which you can see on the left. And you can get as much detail as the images before, so I can ask pretty much the same questions. And I have this lovely video on the right. So this bee actually has a Varroa mite attached to it. So it's actually glued there because I thought that this would help me to locate the feeding site more easily. It didn't, but I still think that it gives you a really good idea of size, just how big these parasites are compared to the rest of the bee. Especially when you imagine that it's not only her in there, it's also her family in the cell, feeding on the bee pretty much all the time. So the more I watch this video, the more I see. So at the bottom of the bee, so at the tail end, there are these 5 fingers that are part of the stinging apparatus. And then just above it on the left, it will just cycle round. There are these 4 flaps that are part of the proventriculus. So the proventriculus moves in and out of the honey stomach, and it's lined with fine hairs that help to filter out things like pollen and pathogens. But it also is part of the system by which the bee takes a certain amount of food for itself to provide energy for itself, and then leaves the rest behind to be made into honey for the rest of the hive. So when I first received these images, I found it quite difficult to believe that they're both pictures of the same feeding site on the same bee, because they look so incredibly different. The one on the left is fluorescence and the one on the right is SEM, if anybody watching does any microscopy. So the one on the left looks how I would expect it to. It looks to me like a deep hole, and it actually to me looks like it's surrounded by an area of inflammation. But on the right we have something completely different, and it took me an awful lot of head scratching and a lot of coffee to find these 5 points that I thought matched up on both images, and it might be a little easier to see it like this. So, like I said, it took me a little while to line them up, but I'm starting to think now that perhaps what I've always thought to be a feeding hole could just be an optical illusion, and perhaps it's more likely to be this partially healed wound site with this outspilling of hemolymph. But I definitely need to go back and get more images like this. But I just think it's more really interesting, and definitely an argument as to why it's important to use multiple methods to look at your work and look at your data. So how does my work affect beekeepers in the field? Well, actually, admittedly, it doesn't at the moment. So there are no direct effects because I'm not studying a different Varroa management method. I'm not designing a new drug. I'm not testing old drugs to see if they'll work on Varroa mite. But what I am doing is I'm adding to a larger pool of information that can be shared by people that could directly affect beekeepers' lives and honeybees. And also it could be of use to people that are studying other insects. So I know that when I'm studying, I read papers that are about flies and about mosquitoes, and that helps me fill in the gaps and anticipate questions that I can ask about the bee. So actually I just want to end on the note that I am not a natural beekeeper. So I started beekeeping as part of my PhD. And actually this is probably, well, definitely the most exciting day of beekeeping so far for me. And I'd received an email from my supervisor, forwarded from the museum, where a lady had been walking in the botanical gardens and she'd seen this swarm that had been there for a while and thought that someone should probably do something about it. So none of my colleagues were available, it was just me. I'd never collected a swarm before, but I had watched a YouTube video and I thought, what could possibly go wrong with the swarm of bees in a public place? But actually I was very confident because swarms are generally very calm. So I went out and I dealt with it. And it was actually very easy, but there's definitely a moment where I check that my veils are definitely shut before I release the bees, because I'm just not quite 100% sure what's going to happen. But yeah, definitely my most exciting day. So thank you so much for listening. Thank you if you've made it all the way to the end. If you have any questions at all, whether that be about honeybees, about my research, or whether you just have questions about what it's like to do a PhD, or the interview process, please feel free to get in touch. I'm absolutely sure that my details are on the website where you found this video. So thank you very much again, and I hope you're all staying safe during lockdown.